The overall goal of this procedure is to enable DNA methylation analysis of CpG dinucleotides by genomic sequencing of bisulfite-converted DNA. This is accomplished by first denaturing the double-stranded DNA so that the bisulfite conversion reaction can take place. The second step is to convert all of the unmethylated cytosines in the genomic DNA to uracil by bisulfite deamination. This reaction involves 3 phases. The first is sulfonation of the cytosine residue by the addition of bisulfite to the 5,6 double bond. Secondly, hydraulic deamination of the cytosine bisulfite derivative produces a uracil bisulfite derivative. And thirdly, removal of the sulfonate group by alkali treatment results in a uracil residue. Next, the target region is amplified by bisulfite conversion-specific primers and PCR amplification. The final step is cloning and sequencing of the PCR product to give single nucleotide resolution for methylation across the DNA molecule. Ultimately, results show DNA methylation of CpG dinucleotides in genomic DNA through exploiting the increased sensitivity of cytosine versus 5-methylcytosine to bisulfite deamination under acidic conditions.
The main advantage of this technique over existing methods like Southern blot analysis is that it can allow single nucleotide resolution of DNA methylation.
This method can help answer key questions in the epigenetics field, such as the role of DNA methylation in human diseases like cancer.
To prepare DNA samples for bisulfite conversion,